So we're going to continue on here looking at the patterns of pronation, the lines of pull, and the muscle cell itself. When we're talking about lines of pull, we're talking about how do each of the insertion points interact with the axis of rotation at the joint, in which the pattern of the fascicle arrangements are going to lead to the shape of the muscle itself, in which we have distinct patterns of pronations that are going to dictate the line of pull that we get from that pattern of pronation, where we will have muscle pattern pronation of parallel, unipennate, bipennate, multipennate, circular, and convergent. And so let's take a look at these lines of pull, and then we'll take a look at how we can get advantage from various ones. So the first line of pull we're going to look at is the parallel. And so in parallel lines of pull, all of the fascicles are pulling along and parallel to the central tendon. So we have one line of pull and they're all parallel to each other. In the unipennate, we have all fascicles arranged at an angle to the central tendon, and that angle is going to vary, but we're only pulling to one side of the pronation pattern. And then as we pull, that angle is going to change, leading to a gain or loss of tensile strength within it. On the bipennate, we still are pulling at an angle to the central tendon. However, instead of pulling on one side, we're now pulling on two sides of the central tendon. And so we're gaining angles and lines of pull within the bipennate that we didn't have on the unipennate. And so far, we've gone from the least amount of tensile strength being the parallel to now having the most tensile strength coming from the bipennate. We now get to the multipennate. In the multipennate, we have varying styles of pronation within the muscle, leading to varying angles within each type of pronation pattern that we have. So what we're doing is we're combining the advantages we get from each of the unipennate and the bipennate in the formation of the multipennate. The last one we get is the convergent. In the convergent, we have an array of lines of pull. There's no true central tendon on the convergent muscle. And because there's no true central tendon on the convergent muscle, we can vary the angle by which the moving end is moving. The convergent muscle provides for not only the greatest amount of strength here, but it also provides for the greatest number of total movements that can take place. And so if we look at where are we going to see these types of muscles, large muscles that are going to be moving things are going to be convergent and multipennated muscles because we need to have the greatest amount of strength there. So things like pectoralis major will be convergent. Things like gluteus maximus will be convergent. Things that need to have extensibility within it but may not provide the greatest amount of strength within it will be parallels, such as the sartorius or the adductor muscles, adductor longus, gracilis. Bipennates are going to be muscles that are going to be providing and performing multiple functions, such as rectus femoris. There's a special case of the parallel and pennate that is a fusiformal parallel muscle, such as what we see with biceps brachii. And so what we have to remember is that the shape of the gaster, the shape of the muscle belly, is going to affect the type of strength that we exhibit from that muscle relative to the lever system of motion based off of the pronation patterning that we see within the fascicle. And the patterning is going to influence the total tensile force, the strength that can be developed within the muscle during kinematic motion, where convergent will have the greatest and parallel will have the least. Now we have to put a caveat on that parallel because circular has no true line of pull within it. And we actually have negating lines of pull in the circular muscle, where circular may be slightly less in terms of contractile strength than parallel. And so the way in which this muscle action is going to work around the joint is going to dictate how we're able to get motion occurring based off the lever systems. When we talk about movements, we talk about the action of the muscles being either a prime motion or an accessory motion. Prime motions are the principal motion that they would use and provide at a specific articulation. Accessory is where the muscle can get pulled in so as to assist the motion. Where muscles are going to be paired around the synovial joint to do one of two things. They're either going to do the motion or assist in motion, or they're going to resist the motion. If they resist the motion, they are antagonist. If they are going to assist or provide the motion, they are primary, agonist, or accessory. The agonist muscle is the muscle that crosses the same joint and provides secondary motion, whereas accessory muscle is a muscle that can get pulled into the kinematic chain so as to provide a secondary means to perform that specific motion. And so now we're going to work into the cell. And when we're talking about the cell, everything gets renamed slightly here, and we're going to call everything sarco. And so all of the normal cellular structures are going to have sarco in front of it, and sarco just is the indication that it is muscle, in this case here, skeletal muscle. And so let's take a look at the various parts of the skeletal muscle. And the first thing that we have to remember is that this is a polynucleated cell. Each one of the nuclei will contain and control a region of the cell itself, known as the myonuclear domain. The nucleus itself will sit just inside of or just deep to the sarcolemma, the cell membrane. The cell membrane has specialized pores within it, which we refer to as the openings of the T-tubules or the transverse tubules. These transverse tubules open up into what's referred to as the triad. The triad is a combination of the transverse tubule. This is an opening of membrane that continues to go through the skeletal muscle itself and allows for transmission of the action potential from the sarcolemma into the cell itself, and the cisternae of the sarcoplasmic reticulum. The cisternae of the sarcoplasmic reticulum is an area where we have sequestered calcium ions that are held there via voltage-gated calcium channels. They run contiguous with the sarcoplasmic reticulum, where when the voltage-gated channels open, calcium is effluxed into the sarcoplasmic reticulum, which will then diffuse into the cell itself, where it will meet up with the sarcoplasm. And inside muscle cells, the majority of the sarcoplasm is made up of the myofibrils. The myofibrils are composed of a host of proteins within the filaments that comprise the myofibrils. The other thing we'll see within the skeletal muscle, and the amount that we see is going to be dependent upon what type of skeletal muscle we see. All skeletal muscles will have a large amount of mitochondria around the transverse tubules and around the neuromuscular junctions. Type 1 fibers and the oxidative type 2 fibers will have large amounts of mitochondria within it. However, the glycolytic type 2 fibers, the 2X's, are going to have very little number of mitochondria within it. And so as we move into the fibrils themselves, we start looking at the actual protein filaments, in which we have two classifications of filaments. We have what's referred to as the contractile filaments, made up of the myosin, or the thick filament, and the actin, the thin filament. Now, we measure them in terms of what's referred to as an angstrom, and that's just a measure of molecular weight. And so notice how the thick filament is larger than the thin filament. Now, we don't say thick and thin filament. We will reference them as their proteins, as actin and as myosin. We also have a couple of regulatory proteins and then some structural proteins that come into play. In terms of the regulatory and structural proteins, what we have is we have Regulatory proteins, troponin and tropomyosin, which are involved with the regulation of the interaction between the actin and the myosin. And then we have structural proteins, elastin, titin, and dystrophin. Elastin, titin, and dystrophin form connective tissues with collagen and with elastin that will secure the myofibrils to the sarcolemma, and the sarcolemma to the endomysium, and the endomysium to the perimysium, and the perimysium to the epimysium, so that when the functional unit of the skeletal muscle contracts, the entirety of the skeletal muscle will be able to contract as a single unit. And so if we look at this, we usually get this quasi-arrangement of structures where we have a central myosin chain held in place by myosin binding protein with its globular kinematic heads, here indicated as cross bridges, surrounded in 360-degree pattern by actin filaments in their G-actin arrangement, covered by tropomyosin, and then regulated by troponin. And so the G form of actin you can think of as strings of pearls. On these strings of pearls, there's binding sites for the globular head of the myosin. And what's happening is a secondary filament that sits in an alpha helical shape will sit on top of that binding site. And what's happening is that there's a regulatory protein that will sit on that tropomyosin, known as troponin, that has a spot for calcium to bind onto it. When calcium binds onto it, it causes a conformational change, and that conformational change pulls the tropomyosin out of the way and will then allow for the actin and the myosin to interact with each other. Within this arrangement, we get a functional unit known as the sarcomere. The sarcomere is divided up into distinct regions. The regions we reference as either zones or bands, and then lines of connective tissue, which we reference as Z lines and M lines. The A band is the length of the myosin. This is the active zone of the sarcomere. The I band is the region where myosin is not present, but actin is present. During contraction, the I band will disappear as the actin is pulled across the myosin. The H zone is the central area of the A band where at rest the actin molecule does not exist. It's absent in that area. The Z line is where the titin protein will anchor the actin molecule together to run in continuation with the rest of the structural connective tissue. The M line is the myosin binding protein that holds the myosin in place so that when contraction occurs, the actin is the only protein filament that will be moving within the sarcomere, pulling everything towards the M line. And so if we look at this arrangement within the muscle, we start with the muscle itself. We then get to the fascicle. The fascicle will have the fiber. Within the fiber, we'll have the fibrils. And then the fibrils will be broken up into their individual fiber units. And so notice how we have this arrangement. Notice the arrangement of the actin and the myosin around each other. They surround each other in a cogwheel fashion. And this cogwheel fashion of actin around myosin allows for this circular spherical unit of proteins to be pulled along based off of action of the myosin globular heads.